it can be determined by many things. It can be determined by environmental factors. If some species that don't have sex chromosomes or have no sex chromosomes that we've identified, their sex is controlled by environmental factors. That could mean population. In some fish, it's population density. It can be pH in the water. It can be other environmental cues like temperature is a common determinant of sex. And interestingly, these things can be superimposed. So in the bearded dragon, there are sex chromosomes. There are ZW sex chromosomes, but that species is sensitive to temperature. So at very high temperatures, dragons that would be males, ZZ dragons that would normally develop as a male, if they are developing at a very high temperature, they'll turn into females. The temperature can override the signal on the sex chromosome in this case. And this has been shown for a couple of species, that even though they have sex chromosomes, environmental factors. In chicken, for example, they have sex chromosomes, they have ZW sex chromosomes, but if you put estrogen on a chicken egg, regardless of its sex chromosome constitution, it will turn into a female.
Interesting.
Yeah. So there seem to be several intersecting pathways.
Yeah.
The system.
So one thing maybe we should do up front here is we've already used some terms that maybe people aren't familiar with. Can you explain the difference for people between gametes, gonads, and genitalia, the 3 Gs here?
Yeah. So, okay, gametes are the cells that are produced that give rise to sperm and eggs. And they are a special cell lineage that is usually formed early in the embryo, and then those cells migrate to their position in the embryo, which is in the gonad, and there they develop into sperm or eggs. The gonad is an organ that arises early in embryogenesis, and it's the only bipotential organ. That is to say, it is an organ that arises and can develop into two completely different organs. It can become a testis or an ovary, the same cells and the same initial little structure. So this is unlike other organs like kidneys. When a kidney forms, the cells that make up the kidney can only become a kidney. They don't have the chance to decide, oh, I'm going to be another organ. And it's true for the heart. But for the gonad arises as this unique bipotential structure, which is what makes it so interesting to study from the point of view of organogenesis. So trying to understand how our early organs form. The gonad is very unique because it can follow one of these two pathways. And the pathway it follows typically controls the sexual phenotype of the organism. So it's the first, at least one of the first, dimorphic, meaning different between the sexes, structure. And once it forms, it begins to produce hormones typical of what it has become. If it becomes an ovary, it'll make estrogen. If it becomes a testis, it'll make testosterone. And then the rest of the body responds to the hormone that is produced by the gonad. So the rest of the animal will be feminized or masculinized by the hormones produced by the gonad once the gonad makes the choice.
I see. So the gametes are basically the sex cells, it's a certain type of cell, like a sperm or an egg.
They're a cell that's produced that gives rise to eggs and sperm, and they migrate. They're called germ cells that form the gametes eventually. But when they're just germ cells, they are also bipotential, and they'll migrate to the gonad. And the gonad has little niches, little compartments to put them in. And then the gonad takes care of those cells. It surrounds the cells in the gonad, surround those special germ cells, and allow them to develop into eggs or sperm.
I see. So the gonad is the special organ that you said it has bipotential. So it can turn into either one thing or another thing. It can turn into either ovary or testis. And it's unique. Right. It's unique compared to other organs like the heart or the intestines or whatever in that way. And I guess what determines which one it turns into is just some combination of sex chromosomes and genetic factors together with environmental factors. And the combination probably varies from species to species.
That's right. But that initial decision within the gonad is referred to within the field as primary sex determination.
I see. And it's primary because it happens early. And then basically you said that then changes the internal environment based on the hormones that get released from that organ.
Yeah. And the genitalia begins as a structure that's also bipotential. It's not really in the same way, but what happens is that it can either turn into female genitalia or male genitalia, but that typically depends on the hormones from the gonad.
I see.
There are a couple of known cases where it depends on the sex chromosomes and not the hormones. That's true in kangaroos and tamer wallabies, so some marsupials. The genitalia development is dependent on the sex chromosome constitution and not sex hormones. It may be that as things go forward, we will find more cases of structures or changes in development that are not dependent on the gonads, but are dependent on sex chromosomes. Right now, we don't have a lot of examples of that, but I think there are some coming along.
Interesting. What about in humans? So what's the primary factor driving whether you develop ovaries or testis in humans? Is that mostly a sex chromosome thing?
Yes, that's the SRY gene on the Y chromosome, same as in mouse. And mice and humans are relatively, or in fact, all of placental mammals are relatively insensitive to environmental factors as far as we know. I mean, they are, because development occurs in the uterus, they are protected from temperature changes, pH differences, unaffected by population density. They're really protected from other factors that really influence the fate of other organisms, for example, that are aquatic, where there may be a high level of estrogens in the water.
I see. So there's this natural buffering there just because of the internal fertilization that placental mammals have. Has anyone done experiments where, in mice, where you just alter the in utero environment in some dramatic way?
Well, the uterine environment is hard to modify. You can't change the temperature. And the placenta basically mediates a lot of the hormone balance to the embryo. Excuse me. But I think that we have tried and others have tried to take gonads out of embryos and culture them in a dish with extra hormones to see whether we can influence their fate. And a colleague of mine, Andy Pask, has found some evidence for this, but we found it very difficult to get any convincing data from this. And it could be that we just that the mammalians, the placental mammalian system is strongly buffered against this. In marsupials, as you may know, only spend half of their gestation period in the uterus. They're born at mid-gestation and then they go into the pouch. When they're born, their front end of the animal is very well developed. They have good claws and when they're born, they climb up the fur into the pouch, dragging the back end of them, which is barely developed at all. I mean, their hind limbs are just tiny at the time they're born. And then they develop for a sort of an equal period in the pouch before they're actually able to be independent. And if you introduce estrogens at the time they're born, when they're born a little early, so you have to catch them at just the right time, but you can influence the fate of the gonad in marsupials.
Interesting.
Suggesting that, maybe mammals are not completely resistant to estrogen, but it's difficult.
Yeah, it's certainly much harder than some of the fish and amphibians.
Yes, for most egg-laying species, particularly aquatic species, if you add estrogen to the water or estrogen mimics, for example, environmental toxicants. There have been many studies, for example, in lakes in Florida, where in the lakes near Disney World, there's a very high incidence of genital abnormalities and defects in alligators and other species that develop in waters where a lot of plastic and yeah, but whereas if you go to some of the lakes deep in the Everglades, you don't see that.